Let's look at how sugars and amino acids get transported throughout a plant. And this is called translocation. So we want to be able to outline the role and the job of the phloem in what we call active translocation of our sugars and amino acids from a source to a sink. So that's all foreign to you right now, but I'm going to do my best to make it something that you understand. So, translocation itself is the transport of products of photosynthesis. So, I mean, mainly looking at sugar, right? The transport of sugar, and this is taken care of by the phloem. And so, when we had studied the xylem and the phloem, we talked a lot about how the xylem transported water, but the phloem is transporting food, and so that food is ultimately we are looking at sugar. Now, transport of water is going to take place toward the leaves, basically going up. And the translocation of food, proteins, and we will see amino acids get translocated. It will go down toward lower parts of the plant. It could go down to the root, it could go down to other cells in storage, but it's going away from the leaves. And the movement of this phloem sap requires energy, and so we have to have active translocation working. So we're going to need some help in this translocation for it to occur. And when we talk about sap, we are basically talking about sucrose, amino acids, some hormones, and most recently they discovered mRNA as being translocated as well. Now, the phloem itself is made of what is known as a sieve tube, and in between each sieve tube is a sieve plate, so it's porous and it's going to allow for the movement of water and molecules. And then next to it we're going to see companion cells, and so we'll either see a source or a sink next to these sieve tubes. And in between, this picture doesn't show it, but kind of what's going on in between is the plasmodesmata, and the plasmodesmata kind of builds a bridge between the sieve tubes and the companion cells so that they can translocate things in between each cell. Now, the phloem itself has a two-way flow, but if that's going to occur, the flow must always be from a source to a sink. So a source, and our example is over here, a source is a net producer of sugar or sucrose, as an example, which has been made through photosynthesis, or if we have broken down starch inside of the leaves. Okay? So our sources are typically our leaves, and you can see the picture back here. We've got a leaf right here and then some sort of root system. So that's our source, mainly in the leaves. And the sink is going to be used to store the sugar. And that could be in, here's our example down here, and that could be within the roots, buds, stems, leaves, or fruits. Now, some of these regions are going to be both a source and a sink. So for example, a tuber or a bulb may be a sink during the summer, and then during the springtime it will be a source. Now, just looking at number one in this diagram, number one at this point we're seeing sugar being loaded into the sieve tubes, and since the sugar has moved into the tube, we have changed the water potential. The water potential has now lowered, and what we're seeing is we're seeing a higher concentration of the sugar inside of the sieve tube. Therefore, water is going to want to follow it through the action of osmosis. We will see positive pressure build within the sieve tube from that uptake of the water, and that's going to force the sap to actually flow along in the tube toward a sink. And so this pressure is going to be relieved once we unload the sugar into the sink, and we actually will end up seeing down here, we will see water shift back over into the xylem, and this is to recycle some of that water. And this is if we're going from the direction of the water going back from a sink to a source. So the water will flow back up to kind of help aid in the process of translocation. And the phloem sap actually moves pretty fast. It's about 1 meter per hour, and so, I mean, that's 3 feet per hour. If you have tiles on the floor in your kitchen, you know, they're usually about one by 1 foot. So 3 feet in an hour, that's That's pretty far, especially if your plant isn't 3 feet. So it's pretty good. The loading of the phloem, just to kind of give you a little bit more about that active translocation, it requires active transport. So in the picture on the right, you can see that we're looking at sort of a symport process where hydrogens on the inside will get pumped through to create this high concentration gradient, and then the sucrose that's going to leave our source is going to be coupled with hydrogen ions and actually move into the sieve tube. The last thing that we can take a look at here is down at this diagram. If there is any sucrose that's made within the mesophyll cells, they can actually travel through the symplastic pathway, and then it can get shifted over into the apoplast pathway in order to get carried over into the sieve tube. So that's just a different pathway that it can take through the mesophyll cells. Here's a couple of videos that you could take a look at, just some animations to kind of review. Probably take a look at them in class as well.